Okay, good evening. Welcome to week 8 of Advances in Omics. So, before I start today's session, I want to say a couple of things. First thing is, I've been getting a couple of emails regarding week 1, question 15 to 17. So, I think when doing our live sessions as well, we had some issues regarding solving those problems as well. So just to confirm for that, so I have asked the person who made the questions for a solution. So he has provided me solution of one of the answers. When I get for the rest of them, I'll be sending those out. So yeah, the questions are a little bit tricky, so I asked him only to give the answers. So he has given one, he'll be giving the others, and then I'll kind of reply to those emails just to be clear. Okay, so another thing, tomorrow at the same time we have an extra session for this course, which will be kind of a recap of the whole 8 weeks, and we'll discuss some topics regarding those. So yeah, that is all I had to say before starting. So yeah, let's get started. So in this week, this is the closing week of the course. So we'll be discussing primarily a little bit into detail about RNA sequencing data analysis, how to do basically transcriptomic data analysis, and the softwares that we use and the kind of algorithms that we use. And we'll be talking a little bit about specific mutations and which cause diseases and so on. Right? Okay, let's get started. Okay, if you remember, so the whole genomic data workflow was that you get the genetic data or the genomic data from the sequencing machine in terms of FASTQ files, and after you get the data, you do alignment with the reference genome, right? So there are a very wide variety of tools in terms of aligning aligning genomic sequences, but alignment of genomic sequences and alignment of RNA sequences are going to be slightly different because of the obvious fact that in case of eukaryotic mRNA or human mRNA, there are isoforms of the RNA, right? So you have the alternative splicing, and it's not a continuous stretch in the genomic region. So what you are doing while RNA sequencing is mostly complementary DNA sequencing, right? So you take the RNA, you use reverse transcriptase to make a DNA sequence out of that, and then you sequence that particular DNA and map it back to the what the RNA sequence might be, right? So when you are sequencing the DNA, you need to align it to the DNA sequence itself, but the whole DNA read basically won't align to the DNA reference genome because it's the complementary DNA of the mRNA, right? So there has been splicing, so intronic parts have been cut out of that sequence. So you need to have an aligner which kind of is aware of splicing and kind of can align a single mRNA sequence to the genome in scattered manner, right? So for RNA sequencing data alignment, one of the most popular used tool is Top Hat, and it kind of aligns the reads in two steps, which is firstly the unsliced reads are mapped to locate exons with a tool called bowtie, and then the unmapped reads are then split and aligned independently to identify the exon junctions, the breakpoints of exons and introns from where the DNA mRNA has been cut. So this is another tool which is HiSat2. It stands for Hierarchical Indexing for Spliced Alignment of Transcripts version 2. It is kind of very widely used for this kind of analysis, and it uses a graph-based approach to index the reference genome combined with the bowtie algorithm for alignment. So it is also important for having quality checks on the reads itself. So you use some clipping mechanisms in terms of control. You use Picard, and other tools are there as well. So I'll just walk through how the alignment strategy works in terms of, so this is the star aligner. This is a tool that is also widely used. So how does it work? So firstly it is the most kind of widely used tool in the field in terms of RNA sequencing data alignment, and it is also done in two steps. One is the seed searching, and second is the clustering, stitching, and scoring. Right. For the seed searching, for every read that this algorithm aligns, it searches for the longest sequence that exactly matches one or more locations in the reference genome, and these longest matching sequences are called MMPs, which are maximal mappable prefixes. Right. So if you have a read, so this is the read. So imagine this is the read which is coming from the mRNA converted to this complementary DNA read, and this read, a portion of that read actually matches to one portion of the reference genome. So the longest sequence exactly matching the reference, these are called MMPs, and there'll also be an unmapped portion of the read as well. And the different parts of the read are mapped separately, and these are called seeds. So the first MMP that is mapped to the genome is called seed 1, and then it will go on for seed 2, seed 3, like number of reads that is present. So what will happen is, after the longest longest match is identified, then it will search again, but only for the unmapped portion of the read to find the next longest sequence that exactly matches the reference genome, which will be seed 2. So if you understand properly, so this is the whole read. Part of the read aligns here, the other part of the read actually aligns here. So basically this part has been cut off from the sequence, which indicates this is basically a intron between these two exons, and these are your breakpoints from which the sequence has been cut. So this is the donor and the acceptor site, splice donor and splice acceptor sites, right? So the sequential searching of only unmapped portion of the reads underlies the efficiency of this algorithm. So again, so if it'll take the whole read, it'll try to map the read as much as possible. If it matches quite a bit, then it'll take the unmapped read and try to match it somewhere else. It uses uncompressed suffix arrays to efficiently search for these MMPs, and this allows for quick searching even against the very large reference genomes as well. So what happens if it does not find an exact match? So if it does not find an exact matching sequence for each part of the read due to mismatches or indels or previous MMPs, will be extended. So what we'll do is, if here is a mismatch, then and you cannot find a match for the unmapped read in the genome anywhere else, so what it'll do is it'll try to clip the adapters or other contaminated sequences, and it'll try to realign with a longer sequence match from the first part. So then when it is done, the separate seeds are then stitched together. The seeds are basically a part of the sequence that you're trying to align. So seed 1 and seed 2. So these are the two seeds, and they are stitched together to create a complete read by first clustering the seeds together based on the proximity or to a set of anchor seeds or seeds that are not multi-mapping. Then the seeds are stitched together based on the best alignment for the read, which is basically they use a scoring matrix based on mismatches, indels, gaps, etc. Similar kind of algorithm that we use for genomic data alignment as well. So this is the alignment part. Now we also talked about genomic data assembly, right? So we can do a similar kind of analysis with transcriptomes as well. So if you do not have the reference genome for these transcriptomes, you need to kind of do an assembly, de novo assembly, right? So a transcriptome conducted from short read RNA sequencing, this is a workflow for that. I'm muted. Okay, so a transcriptome, this is a transcriptome constructed from short-read RNA sequencing, uh, and it's an easily attainable proxy catalog for protein coding genes when a genome assembly is unnecessary, expensive, or difficult. So if you just want to find the protein coding genes sequences and you only have transcriptome data, you do not need to do the whole genome assembly. Transcriptome assembly like this would be enough. In the absence of a sequence genome to guide the reconstruction process, the transcript must be assembled de novo, only using the information available in the RNA sequencing reads. Subsequently, the sequences must be annotated in order to identify sequence intrinsic and evolutionary features in them. For example, protein coding regions, and although straightforward at first glance, de novo transcriptome assembly and annotation can quickly prove challenging undertaking. So what they do is they take this, so these are divided into these 6 steps basically. So first what they do is take the raw reads and they kind of remove the adapters, the contaminations of other RNAs like tRNAs or other small RNAs that are there. They remove those contaminations. Then they do uh, mapping and abundance estimation. So basically similar type of reads, how many are present, and like what is the total number of reads present for a particular read. So one particular read may, if there are 15 times, 14 times, 10 times, etc. And what it means is the more number of reads that is present, it is directly proportional to the expression of the transcript in the cell. So it is directly proportional to the gene expression. So if you do not even have the annotations of these transcripts, what you can find immediately is basically what is the quantification differences in terms of different transcript levels in the cell. So if you have if you, for the, so basically if you want to know what these reads actually represent in the gene, like what, from which gene these reads are coming, then you need to do the whole assembly part and understand the annotation and do the statistical testing. So what you do for de novo assembly is you kind of do the similar type of an algorithm that you try if you have multiple transcript reads that you have kind of overlapping sequences with. So you do you construct a graph and try to do a whole assembly procedure, right? So then you try to classify if you can understand which are mRNAs, which are tRNAs, rRNAs, so on, and if you still have any kind of extra RNA sequences which are not mRNA. So then you annotate them according to databases. So you have transcript sequences in certain databases that you can know that these sequence actually refer to these reads. So you can do the annotation and you can go back to the statistical analysis that these many reads are coming from these many genes, and like it is more or less, or in terms of statistical testing you can do that. So similar type of concept like genome assembly. So yeah, this is a little bit in detail about how they do that. So it's the similar type of exercise where you construct the k-mers and then you construct a De Bruijn graph and try to find the shortest possible where you're trying to find the whole sequence from the graph itself, right? So yeah, so the basic idea is to establish a catalog of substrings from the RNA sequencing reads, compose them into a set of graphs wherein substrings are connected if an overlap between them exists. This establishes paths through the graph which correspond to the transcript reads potentially that have originated from them. It's a similar thing. I'm not going into the same details again, but yeah, if you remember the genome assembly discussion, it's quite similar here. So now you have quantified the in terms of you have identified the transcripts, which genes they are originating from, and so on. Now what you want to do is try to quantify the transcripts amount in the cell or in your data, right? So it is a fundamental task because that will tell you how the gene is getting expressed inside the cell. So so in case of RNA sequencing, the features. So let's go from here. So accurate and efficient quantification of transcript abundance from RNA-seq data is an especially pressing problem due to both the wide range of technical biases that affect RNA sequencing fragmentation, amplification, and sequencing process, then exponentially increasing the number of experiments and an adoption of expression data for medical diagnosis. In the case of RNA sequencing, the features are typically genes, where each gene is considered here as a union of exons. Again, so for that to know that all the transcripts that have come from a particular gene, you need to do that alignment part, right? Otherwise, you won't know. Or kind of annotate the sequences of the reads to particular genes. So when you're talking about a gene, it is actually union of all its exons, how many transcripts that is present. One may also consider each exon as a feature in order to check for alternative splicing. And for comparative ChIP-seq, the features might be binding regions from a predetermined list. So you can do it exon wise, you can do it in terms of binding region wise. So there's a room for for people to explore in terms of how they want the data to be quantified, right? Special care must be taken to deal with the reads that overlap with more than one feature, and these are basically softwares that help with it. The HTC count is a software for that. Yeah. Uh, okay. So, sorry. Yeah. HTC count is a software for that. So the three overlap resolution modes of HTC count works as follows. For each position in the read, a set S is defined as a set of all features that are overlapping that position. So you have one position, and then you take all the transcripts that are as a set that are overlapping that position, and then consider the set S, which is basically with I running through all the position within the read. Firstly, the union of all sets SI for mode union. Then you have intersection. So either you take, so for that point, do you take all the reads that are kind of mapping in and around that region, or do you take strict intersection of the reads that are present in that particular position, and also intersection of non-empty sets for mode intersection non-empty. So these are the different modes, right? So if S contains precisely one feature, the read is counted for this feature. If it contains more than one feature, the read is counted as ambiguous. So the mapping of the read needs to be non-ambiguous to be counted as a feature, and if it is empty, that there is no feature. So let's take this example. So you have a read here, the gene is spanning here. So it is if you take union, intersection, and intersection that are non-empty. So for all this case, it is mapping to gene A, right? So if you take union for the second case, it is still mapping to gene A, but it is not fully mapping to gene A, right? Some of it is unmapped. So if you take intersection strict, it will give you no feature, and if you take non-empty, it will give you back gene A. So here also it's a similar condition. Half of it maps to one exon, half of it maps to some other exon. So the union will be again gene A, no feature for intersection because it does not wholly match with the particular exon. Non-empty is basically gene A. So you have this kind of uh, very different scenarios that can arise due to this alignment thing. So you have, in this case, it is fully ambiguously aligned. So you have the read here, which is kind of overlapped between gene A and gene B, and you do not know from which gene it actually originated, right? So it is given as ambiguous. It is not quantified in terms of the transcript numbers. So this is another algorithm which is known as RSEM, RNA sequencing by expectation maximization. This is a very famous algorithm that we use in other spheres of genomics as well. So this implements quantification method and provides extensions to the model. So a key feature unique to
Excuse me, sir.
yeah.
Sir, sorry for the interruption. Because of my academic schedule, I am unable to attend the problem-solving sessions. So I
Yeah,
used to watch recording of the session, sir.
yeah.
Uh, so that I am facing difficulty in solving the problems. Some of the problems in week one. I also mailed you
Yeah.
about this.
I don't know when you joined basically. When due to so when I started this session, I basically told that I've gotten a mails. I think a couple of others have also mailed me for the same issue. So question number 15, 16, and 17 for week one. The thing is, the
Yes, sir.
questions are a little bit trickily worded. So I did not want to give my since I have not met the question, I did not want to give my solution. The person, the TA who has made the question, who is actually a Sir's TA, Professor Nagarjun's TA. So I've asked him to provide the solutions. He has given me solution of question number, I think 16, the middle one. The other two, 15 and
Okay, sir.
17, he has not given me. The moment he gives me, I'll forward it to you. And if you want, by the end of this class, I can forward you uh, the solution of question 16 right now, and when I get 15 and 17, I'll forward it to you then. So whichever works for you.
Okay, sir. Thank you, sir.
Yeah.
Because of some club activity, I am not able to continue the class, sir. Shall I leave?
Yeah, sure, no problem. This is recorded. If you want to see it later, it will be there.
Thank you so much.
Okay. Okay, so we were talking about this algorithm RSM. So basically, this is also, I'll not go into very details of this algorithm, but this is also used to kind of understand read positions of the particular reads which are mapping to multiple locations. So it uses maximum likelihood abundance and expectation maximization algorithm for the model, and this is a very also quite widely used algorithm in terms of RNA sequencing data alignment. The next thing is normalization, which is very important because so we were talking about quantification here, right? So here we were talking about how many copies of a particular transcripts you are getting in terms of the reads. In terms of the read, so you are getting like say one read having 15 copies, one read having 10 copies, and tell you the gene expression of the of that particular gene in the cell, right? But the idea is that it is not um, without biases, right? So if, for example, imagine the gene is very big, so due to its normal expression in the cell, under that condition only it will produce lot more transcripts than a small gene that is getting over expressed in the cell. So there needs to be control for in terms of gene length, in terms of in terms of the total number of transcripts that are coming in from that particular cell, particular transcript, and so on. So the normalization procedure in terms of RNA sequencing is very important, right? So so what the conditions of the experiment remain is basically sometimes you have a control and then you have a set of cases, right? So control 1 versus case 1, control 2 versus case 2, like that. So you need to have a comparison between control and case to find out which genes are getting over or under expressed. Yeah, so that is the whole purpose of a experiment design for RNA sequencing, that you can compare between different conditions to understand which genes are getting differentially expressed. So for that you need to start from the same position. Should not have biases in your data, and things like gene length normalization, and in terms of transcript quantification, normalization is very important to kind of compare between different conditions, right? So here is an example. So the number of reads aligned to a given gene in a given sample is generally considered a random variable, and so read count comparisons must take into account the variability of these random variables. As I was saying, the gene length might be a variable thing. Second, the total number of reads can vary across samples, so large difference in the gene's read count between different conditions may simply be the result of differential coverage rather than differential expression. It is the second problem that necessitates normalization of read counts before differential expression analysis can be performed, and it is an essential step in terms of RNA-seq data analysis, in which the read count matrix is transformed to allow meaningful comparisons across samples, right? So yeah, so here is a good example in terms of if you don't do normalization, what can happen, right? So one this is a highly expressed genes, and an experiment is performed with the conditions A and B to compare expression for three genes, 1, 2, and 3, gene 1, gene 2, and gene 3, across two different conditions, condition A and condition B, sorry. So in the first section, gene 3 is twofold upregulated under condition B, while the other genes are not differentially expressed, right? So if you just look at the bar plots here, so under condition A, these are the gene expression levels in terms of mRNA, number of mRNAs per cell, that will tell you the gene expression. So under condition A, gene 3, and under condition B, gene 3, if you compare this, condition B, you have almost twice, twofold upregulation in condition B, right? The quantity of mRNA is same for genes 1 and 2, but is twice as high for gene 3 under condition B, which is given by in this particular bar plot. So if you see in the first condition, 1, 2, 3, this is the proportional shares of the mRNA of the three genes combined. So you have equal proportions in condition A, and if you have more proportions in terms of condition B, right? But Let's move on to the next condition. Because of the change in the expression of gene 3, the shares of mRNA in the cell are different between conditions. Under condition A, each gene gets one third, whereas under condition B, gene 3 gets half, while the other gets the one fourth, right? So in condition A, it is aligning to reads aligning to one gene is given one color, so it is equally aligned in terms of the alignment of transcripts. In the second case, it is basically the gene 3 is, in terms of alignment, has higher numbers, and gene 1 and 2 in terms of in terms of alignment have lower numbers. But if you look at the first condition only, if you do not look deeper than the first condition, it only looks that gene 3 has more expression. But in what the reality of the case is, because that is the case because you have more reads aligning to gene 3. But if you look at the proportion of genes 1 and 2, the quantified read aligned to genes 1 and 2 has decreased. So since the total number is constant, in terms of gene 1 and 2, it is decreased. So differences in shares of mRNA are reflected in the shares of shares of the reads. Each sample has the same total number of reads, but the distribution is different under different conditions, matching the distribution of mRNA in this case, right? So when no normalization is performed, there are apparent differences in read counts for all three genes. Total count normalization produces the exact same result as no normalization at all, as total read count for each sample is the same. In truth, there is no difference in expressions of gene 1 and 2, and the relative count for gene 3 should be higher than found by no normalization on total normalization count. So if you look at no normalization, this is the condition. So you have more reads aligning to gene 3 if you compare condition A and condition B. Under no normalization, you have more reads aligning to gene 3, which is given by this bar, and you have lesser reads aligned to gene 1 and 2, given by the smaller bars. This is what happens if you don't do the normalization, but this is what you will see. But the real story is kind of a little bit different. If you do normalization in terms of total count, if total count of the mRNAs, this is what it happens. So there also you do not see any difference, because the total share of the mRNA is still 1. So you're basically, what you're normalizing by is the total number of transcripts from gene 1, 2 and 3 in both the cases. So no normalization and total count normalization do not give you any difference as such. But in truth, there is no difference in expressions for genes 1 and 2, and the relative count of gene 3 should be higher than found by no normalization or total normalization count. Correct normalization therefore makes the read counts of the non-differentially expressed genes equivalent, which also makes the relative expression of gene 3 correct. So no normalization and total count normalization will fail to equilibrate the read counts of the non-differentially expressed genes, resulting in each gene appearing differentially expressed, while the truly differentially expressed gene is in condition B is gene 3. So if you don't do normalization, you'll see something like this, right? In this case, you are seeing that the gene expression level of these two have come down, but gene expression level of gene 3 has gone up. But when in the real case, if you do the correct normalization, this is what it turns out. So normalization, again, just to emphasize on the fact that normalization is very important in terms of RNA sequencing data. If you do not do the normalization correctly, you will be getting spurious results, you will be getting hypotheses, you will be getting patterns that actually do not exist. So that is very important. So moving on, types of normalization that can happen, there can be two steps. One is the within sample normalization, and other is the between sample. Within sample is the same experiment, you are normalizing across that, and between sample is you have two conditions, you have two different basically samples that you are comparing, and you need to put them on the similar footing to make that comparison. So that is the between sample normalization. So within sample normalization helps us to correct the expression levels of each gene associated with the expression level of other genes in the same sample, right? So as I was saying, if you have a larger gene, by default you'll have more transcripts. If you have a smaller gene, by default you'll have lower transcripts. That has nothing to do with the expression levels of those genes. So you need to correct for these things within sample, and between sample you need to remove any batch effects that has, let's say for example your first experiment on the first sample had lower coverage, your second experiment on the second sample had higher coverage. So you'll see automatically, if you don't do the normalization, you'll see that B genes are getting expressed more than A genes, but that is not the actual story. The actual story is that the coverage is more there. That is why you'll see it seeing more expression, and here the coverage is low, but the expressions are more or less similar for except for a few genes. So that is again very important. The most widely studied methods for within sample and between sample normalization are these two terms, RPKM and FPKM. So one is reads per kilogram million, reads per kilobase million, and fragments per kilobase million. So basically you divide and then you convert it into per million, right? So however, the different gene lengths can lead to a bias in per gene variance for differential expression analysis of low abundance genes. In literature, within sample normalization approaches for RNA sequencing data have corrected the biases arising from library size, gene length, and GC content. So these are kind of confounding factors that can be possible. So again, I'll not go into much details of these quantities, but you have these two particular methods, these two particular kind of formulas to quantify in terms of per kilobase of exon per million reads mapped. So this is the normalization. So you first normalize by the number of exons, then the length of the exon, and then you quantify it in terms of million reads mapped, right? So this is another one in terms of RPKM and FPKM. This is transcripts per million, which is basically the sum of all TPM values that is the same in all samples, such that a TPM value represents a relative expression level in principle that should be comparable between samples. So that is transcripts per million. So that is also a value that is used to compare across different samples. So other than that, there are other count normalization methods. This is one of them. This is an R package called TexImport. This used count level data for these RSM output files that we discussed, and the normalized count data were derived from the DE-Sig2 package, and you can also do some Z-score normalization on the TPM level data as well. So yeah, I'm not going to all the details of this, but just to give you an overview about some things that some normalization techniques that are widely used. So, but there are some drawbacks of these normalization. So firstly is basically when there is poor performance when transcript distributions differ between samples. These can perform poorly. So if transcript distributions itself are differing, so in terms of coverage, as I was saying, that can be one of the reasons. So that in that case, these kind of normalization techniques probably won't work very well. And there can be also residual variability from non-desirable experimental problems that are not easy to identify or correct. So there can be, even if after you take all these normalization steps, there can be things. No, I don't think so. No, I'm just that's why I'm not going into details. I'm just giving you an overview about how the data is processed. I don't think you'll have questions regarding these formulas. You just focus on what has been taught in the regular course weeks and the assignments. I think you should be fine for that. These are like kind of extra things that I just wanted to give you, so that if you like in the future become interested to do things like this, these things will be helpful then. Yeah, okay. So yeah, I'll skip these. So much normalization, I don't think it is required. Yeah, so one thing that is again very something that is kind of very widely seen in are batch effects. So it is variability due to differences in terms of just give me a second. Hello. हाँ. क्या? एक सेकंड, मैंने नहीं देखा. मैं देख मैं अभी क्लास में हूँ, तो मैं तुम मुझे टेक्स्ट कर सकते हो। Yeah, sorry for that. Sorry for that. Okay, so something that we see is batch effect removal, right? So the variability in terms of uh, in terms of the data, not because of differences in biological expression of genes, but differences in experimental conditions. So say for example, a sequencer gives you, in terms of data, they give you some variability, and the other sequence gives you some other kind of variability, and when you try to merge the data, you see these data are not clustering together even they are from the same condition. They're clustering separately in using techniques like PCA, and then you see that the variation is not due to the biological approach, it is biological phenomena, it is due to the experimental approach, right? Due to experimental design. So what you do is basically try to remove the batch effects. That is what is called. So for example, you have here three batches. Batch one is the red dots here, data sets, samples you can assume here. 2 and 3 are these two other batches which have blue and green uh, blue and green colors for the samples, right? So for the red ones, when you do a clustering, it clusters out separately compared to the batch 2 and batch 3, because it is not because of the variability in terms of the data itself, but it's the variability uh, of the experimental design. So you use some stuff to remove to remove the effects of this experimental design. So these are common approaches, which is lemma and combat. So lemma is quite widely used. It uses a two-way ANOVA approach, and this combat uses an empirical base approach, which is critical for small batches to avoid for overcorrection. And after correction, it looks something like this, right? So they here the clustering has occurred over a larger area, but it is overlapping with the other batches. So it tells you that the variability, still the variability is there, because these are the green dots, here are the red dots, so they're distinct in terms of the samples biological information, but the variability in terms of the uh, experimental design, that has been removed. So that is the whole concept of batch effect removal. Okay, so not going to these. These slides will be there if you, in the future, if you want to take a look, you can take a look. Quantile normalization I'll not go into, but yeah, let's go and look at some downstream analysis. So what they do is, I think this has, okay. So after you've gotten the data that these are the transcripts mapped to these are the genes, this is the normalized uh, quantified value of these transcripts, and then what you try to do is try to cluster, as I was talking about in this particular example, the PCA thing. You try to cluster these. So you see, if you cluster the samples, you'll see the control samples grouping together, the one of the cases grouping together, and the other cases grouping together. So this is kind of uh, expected in terms of gene expression analysis. So the test that you do to understand if a particular gene is differentially expressed or not is mostly student t-test, because you are doing that normalization, you are doing z-score normalization, fpkm, rpkm, whatever you are doing, you're converting into a normal distribution, so you can use student t-test for that. So this is quite straightforward, that you calculate the signal by noise ratio, which is between group variability and within group variability. So this is the difference it quantifies, right? So In some cases you cannot use it is the one where the distribution is not normal, because t-test assumes that your distribution is normal. Since you are doing so much normalization, the distribution should be normal, but in some cases it is not, so that there it can be an issue. So yeah, it is good, but modern softwares like EJA, DESIC2, Lima, they use some other variance estimators. So I'm not going to details, but again, there are some issues with t-test, that some caveats that you need to know before you are delving into a differential expression analysis using t-test. So there are other softwares that can do it in a better statistical manner, right? So not going to details, so you can use, these are linear models basically that they use to um, kind of do the analysis. So there are some softwares that you'd want to remember. So CovDiff2 is a major one that uses these GLM methods to understand differential expressions of genes, right? So let's take an example of here about how it does it. So this analysis is done with DSIC2. So is kind of the final step of the whole analysis pipeline, that you've gotten the reads, you've annotated the reads, you've quantified the reads. Now you want to see how they are differentially expressed. So the first step in the differential expression analysis workflow is fitting the raw counts to a NB model and performing statistical testing for differentially expressed genes. In this step, we essentially want to determine whether the mean expression levels of different sample groups are different or not. So it's quite simple. So you have two groups. So blue ones are similar groups, red ones are the other groups, and it, you want to check whether there are significant differences in terms of the group means. Now, if you only take the blue dots, this is the mean of that particular group. If you take only the red dots, this is the mean of that particular group, and if you take the whole data together, this is the mean. So there are significant differences, right? So in this case there are, but if you check in this case, they are mostly overlapped, and the group means, the global mean is not very different from the individual group mean. So that also becomes a problem, that there is no statistical differences. So if that this was a very wide, very kind of zoomed out overview of what it does. But if you look into the paper, uh, it uses generalized linear models from the EJAN methods that we just gave an example about. So it involves multiple tests. So this is the flow chart. So you take the read counts associated with the genes, you do the normalization, clustering, and then you model raw counts for each gene. You do some statistical stuff here. So first you estimate the size factors, estimate gene-wise dispersions, fit the curve to gene-wise dispersion estimates, shrink gene-wise dispersion estimates, and then you finally find a GLM that fits for each gene, and then you calculate log to fold changes, and then you test for differential expression analysis, right? Yeah, this is kind of the workflow that it works on, and it kind of helps with complex design, so you do not need to worry too much about that. So if you want to, say for example, use confounding variables in terms of age, sex, some treatment conditions that is there for the samples itself, you can provide that in the design, and the model will take care of it. So here is an example of it. So here basically, these are the sample IDs. Then you have the sex of the samples, the age, the treatment, and the treatment of treatment sex, which is sex treatment, which is basically an interaction term, right? So again, these are different confounding variables that you can adjust for while comparing two different samples gene expression levels. So again, I'll not go into very details, but yeah, so if you want to look into the details of DESIC2, you can look into this PPT. I'll upload this PPT in the same place as we do for the other ones. If you want to take a look, you can go ahead in the later class. I'll not go very deep into this because this is not part of your syllabus. I'm just trying to tell you the things that kind of are in use. Okay, so yeah, let's move on from this. These are all the steps that the DESIC2 model uses. Okay, now let's move on to some examples and in terms of the gene protein interactions. So in terms of gene protein interactions, there can be various types of interactions. So here are four kind of interactions shown. So a particular gene getting a particular protein which is involved in some reaction, then other similar genes, so different genes is giving different types of proteins. Or in terms of proteins, and you can assume these are mRNAs, involved in a particular reaction, or you have the reaction you can assume as different biological cellular processes, right? And then you have two genes giving similar product which is involved in some interaction, and then you have one gene giving one product which is involved in multiple things. So these type of interactions are possible. So this example is given in terms of enzymes, right? So you have, these are what are known as isozymes. So same type of enzymes that can catalyze a reaction. This is what is known as a multimeric protein complex, where you have one gene uh, producing one part of the enzyme, the other gene producing other part of the enzyme. They come and bind together to form the whole enzyme, and which kind of is the functional one. And then you have multifunctional enzymes as well. So one enzyme catalyzing multiple reactions. So this analogy can be done in terms of genes and phenotypes as well. So if you can assume this is the gene, this is the mRNA, this is the phenotype. So this whole conditions actually work in those conditions as well. Now, in terms of protein function, so we talk about two major things in protein function. One is the loss of function, another is the gain of function. Yeah, two different types of mutations, major types of mutations that we look for are loss of function and gain of function. So the loss of function, which is firstly, it can be complete loss of function, that the gene is not functional at all. So it results in a complete absence of the protein or its function. The second is reduced function, which is known as a hypomorph. The previous one is known as a amorph. Hypomorph is basically reducing the protein's activity or the amount produced, leading to a partial loss of function. It is still functional, but the function has been toned down a lot. So that these are two different types of loss of function variants that you can have. In terms of gain of function, it one is hypermorph, which can like kind of enhancing the normal function. With another one is new function, which is neomorph, which it gains a novel function in terms of what it was doing previously versus what it is doing now. So it can acquire a novel function or is expressed in a new location, which is ectopic expression. The other one is interfering function, which is antimorph. This is known as a dominant negative. A mutant protein interferes with the function of the wild type protein. So you have two copies. So in one copy you have a gain of function, one copy you have the normal protein. The gain of function protein actually kind of halts the function of the normal wild type protein. So it's kind of dominant negative. So you do not ultimately have do not have any function for the gene. The distinction between loss of function and gain of function is not always super clear. Loss of function usually means the less of a protein is made, or that some function of the protein has been compromised. Loss of function mutations are usually recessive. This is important because in terms of evolutionary selection, if it is a homozygous mutation, then then even you do not have one copy of the gene to be functional. Generally, that person or that being or that species would not survive. So, in terms of selection, loss of function mutations are mostly under recessive conditions. And since in most cases a single good copy of the gene will suffice, as if you have a good copy, it will work well. So it is still possible to have loss of function mutations as a heterozygous copy or in a recessive mode. Two common types of exceptions are haploinsufficiency, which is the condition is that one copy is also not enough. You need both copies to function. So even then, so in that case, even if you have a recessive mutation for a loss of function gene, for a loss of function mutant, you still have a complete loss of function because protein function will be compromised because one good copy is not enough. And the other exception is dominant negative, or as I was telling, the anti-morph condition, where the defective gene interferes with the function of the wild type copy. This is common with proteins that form polymeric structures such as filaments. So, coming from filaments, this is a very widely known disease that has been studied for a while, which is cystic fibrosis. It shows the expected recessive pattern of inheritance of a loss of function allele of a gene. This is this particular gene, CFTR. It is a cystic fibrosis transmembrane conductance regulator. It's the whole name. It is a salt transporter required for normal function of the lungs, pancreas, and other tissues. It is a large gene that encodes for a large protein, making it a big target for mutations. So again, larger the gene you have, the more mutations, probable mutations you can have in that particular gene. Another example of a recessive loss of function allele is lactose intolerance, which is usually a result of a reduction of function alleles that have low expression of the lactase enzyme in adults. So this is a very interesting example. So this particular lactose intolerance phenomena is from a historic perspective. The mutant form is also is very common because in case of most mammals, including early humans, we did not used to drink much milk after infancy. So the lactose gene was inevitably inactivated due to selection, right? But in many human populations, particularly in Europe, where dairy cows were domesticated, acquired the ability to metabolize lactose even throughout adult life, most likely by mutation of regulatory elements in the lactase gene promoter region. This has apparently happened independently among some East African populations as well. And this lactose intolerance, due to this whole historical prevalence, it is very prevalent among non-Europeans, because Europeans actually developed uh, the mutation that got selected because they wanted to metabolize lactose higher in their adult life. So from an evolutionary perspective, the mutant got selected as well. So it is very prevalent among non-European populations as well. So lactose intolerance is dominant over intolerance. Sorry, lactose tolerance is dominant over intolerance for reasons that are obvious, because we drink milk now a lot. In other words, lactose intolerance shows recessive inheritance. So this is another good example of that. Another good example is another disease called Marfan syndrome, which is caused by dominant negative mutations in the FBN1 gene. It encodes for a protein called fibrillin 1, which forms microfibrils in the extracellular matrix. So these are the conditions. You have kind of deformed hand and leg structures because of the gene in this mutation in this FBN1 gene. So this is an example of a dominant negative mutation as well. And one last example is of haploinsufficiency, that when you have one copy of one wild copy, one loss of function mutation, but that one wild type copy is not good enough. So this is a very widely studied phenomenon, which is the familial hypocholesterolemia. I kind of also work with these genes as well, LDLR. So LDLR is basically the LDL receptor in the cell, right? So LDL is a type of lipid that is metabolized by the cell. So when you have LDL, you have a receptor that kind of carries and does some signaling, and which carries it to the endosome, to the lysosome, and forms into cholesterol. So that is how LDL is taken up from the blood, right? So loss of function mutations in the LDLR gene is basically very harmful, because even in a recessive condition, it causes haploinsufficiency, because if you do not have two copies of the LDLR gene in wild type manner, it won't work. So lots of different mutations cause dominant familial hypercholesterolemia by disrupting the LDL receptor function. There are a lot of, this is the whole exonic structure of the LDLR gene. Then you can have a lot of type of variations. So the variations are given by these legends. So you have frame shift, here. So there are a lot of variations that can happen in different exons of the of the protein. So these are different exon numbers, and these are different functions of these exons, and can get affected anywhere, and this causes actually haploinsufficiency, which is familial hypercholesterolemia. Okay. So the last example here is the gain of function. This is also a very interesting example. This is also very prevalent in India as well. So I hope all of you are familiar with the hemoglobinopathy, which is sickle cell anemia, where you have where your red blood cells goes from here, which looks like a normal cell, to goes like sickle shaped, which has less surface area and less ability to carry oxygen in the body. So this is called by a single point mutation in the cell, in the gene, and in the variations in the beta globulin gene. So this is the beta globulin gene, the HBB gene, that is affected, and it causes the sickle cell anemia. The disease is inherited in a recessive trait, but the same mutations result in a dominant inheritance to resistance to malaria. Now think about it carefully. So what is malaria? Malaria actually is a disease caused by a virus. If I'm not wrong, Plasmodium vivax. If I'm not wrong, I might be wrong on that, but it affects the hemoglobin cells in the body, right? So if you do not have normal hemoglobin cells, the it becomes harder for the pathogen to attack that type of cell and cause the malaria. If you have sickle shaped hemoglobin cells, it is much harder for the pathogen to infect your hemoglobin cells and cause malaria, right? So that sickle shaped cell causes a disease which is sickle cell anemia. You can like suffer it through your whole life, but it also gives you resistance to malaria, because malarial infections cannot take place with the sickle shaped mutations of these hemoglobin cells. So it from a logical perspective, it makes perfect sense. And so the idea is that this disease of sickle cell anemia is inherited as a recessive trait, but if you have this particular mutation, you are kind of dominant negative towards malaria. So you have dominant inheritance of resistance to malaria. So even if you have one copy, that is enough. So that is also a very good example of gain of function. But the gain of function again is with respect to the specific function. The same mutation is causing a disease which is sickle cell anemia, but the same mutation is giving you a resistance to malaria. So it is gain of function in terms of the malarial resistance, but it is kind of a loss of function in terms of the sickle cell anemia disease. So this becomes a very good example of that. Okay, so that's it in terms of my discussion part. Now we'll move on to the assignment questions. Before that, if you have any questions, you may ask. Yeah. Which past assignments? Okay, can you tell me which week is this? Week folder, let me pull it out. Okay. Number of Okay, if I remember correctly, this is the question. I think you're saying the sequence length is 36, 35. Okay. I think it is 36. I'm getting 36. Okay. So the idea was that the pi is calculated by the basically the pairwise weighted difference divided by the total ordered pairs times the sequence length. So that was the formula that we applied, and I was told the sequence length is 36. I think it is 36 from where I'm counting. Just let me count it again. Okay, 32, 35. Okay, I thought it was 36. 1, 4, 8, 12, 16. Okay, yeah, I can see it. Yeah, probably. Okay, so let me go back. I think this is what we are looking for. So if you're trying to find the Tajima's D, the D is given by so this is the formula. So difference by the variance, right? So the difference is between the average pair of nucleotide differences and the expected diversity from the number of segregating sites. So this is your average pairwise differences, and this is the expected differences, which is basically all the combinations of the differences that are possible, and then you divide it by the variance of that site. So in terms of when you calculate the pi, it will be where did it go? Now when you calculate the pi, it is going to be the pairwise differences, pairwise differences, and then you divide it by the total possibilities. So the ordered pairs, total number of ordered pairs is 380, and in this case I thought the sequence length is 36, so 35. So even with this, is it not coming? So let me try once. 537 divided by 3 times 6. Okay, it's coming a little bit more. Okay, let's see that I have not made this question, so So these are some, like, they have used some tricky wordings with this, so I think I'll have to kind of ask them only, the people who have made the questions. Just noting it down. I'm going to write an email today only, to sir and to his TA, because yeah. So this is week 4. I'll have to get back to you regarding this, because I was told the sequence length is 36, so I did not do so much of digging on this. 16. I'll have to get back to you on this. I'm not pretty sure. Yeah. Okay. Let me bring it up 1 second. Week 2. Which is this question number? Can you please tell the question number? I'll try to look for it. Question 2. Question 2 is something else, I think. Just a second. Is this the question you're talking about? This is week 2, question number 2. Then which is the question number? Because this is 2, this is 1. NPTEL assignment. See, I have access to older assignments. 1 second. Yeah, so week 2. This is question number 2 in week 2. NPTEL assignment, is it something else other than the week assignments? Oh, wait, good question. DDNTP is dideoxy, right? Mm, one second. Okay, so you have DDNTP. I think you won't. Mm, mm, okay, so if you have dideoxy, you won't have anything on the uh, sugar moiety. Uh, you won't have anything on the nitrogenous base as well, but phosphate you'll have PO4OH. Okay, I think it should be 4. What is the correct answer? Yeah, single isolated DDNTP. Okay, guys, I do not have access to that question, but I think it should be 4, because you won't have anything on the sugar, because both your dideoxy are like. Have you raised this in the discussion forum? I think these assignment related things you should raise on the discussion forum as well, because their sir sees and his TA sees as well, so they can help you better with this, because they are the ones who have made these questions, right? So yeah, I think it should be 4, but I don't, I'm not completely sure, because there might be something like tricky here. So I'm thinking of in terms of isolated DDNTP, so something with isolated, but still it should not be 5 from my understanding. Mm, okay, sure, guys. But can you tell me the question number for this, and which week, and what is the, this is a separate assignment other than the weekly assignments? Oh, okay, okay, okay. I don't have access to that. Okay, but still I'll question 2. Yeah, I'll just copy these questions. Oh, 1 second. Okay, I'll raise this things with sir. Not sure. Okay, along with these questions 15 to 17 from week 1, I'll raise these as well. Okay, I'll try to bring the solutions. I'll ask sir. Okay. Okay. In second generation sequences, base quality versus base quality versus sequence cycle. Okay, I think it should be, Yeah, option 2, if I'm not wrong. It's an increasing curve. So the first derivative should be positive and the second derivative should be negative. Which is the correct answer? And which week is this? Week 1. Okay. Question number? Let me check. Week 1. Week 1 12th question. Okay. I think we did not discuss this in the session because it is not in my PPT. Let me see one thing. Okay, so if that condition is correct, it should be somewhat like a hyperbolic curve, if I'm not wrong. So you have first derivative less than 0 and second derivative less than 0 as well. So it is somewhat of a something, or just let me try to plot it. Yeah, I think it is correct because if you look at the curve that happens, sorry, something like this, right? So you have an increase, most of them. So let's talk about something like this: per base sequence quality versus, this is position, but I think for sequence cycle also it should be like this only, if I'm not wrong. So if it is a concave curve like this, the first derivative and the second derivative both will be negative. Yeah, if you assume something like this is not this is per base sequence quality, but I think it will be similar. But you have a concave decreasing curve. Yeah, you need to have a concave decreasing curve. So at every point your uh, yeah, so it should be like something of a y equal to negative x cube type of thing. So it starts from here and it goes down. So if that is the curve, the first derivative will be negative because this is decreasing. The second derivative will also be negative because that will be the maxima, right? So at this point it will be maxima. So the second derivative and the first derivative both will be negative if it's something like y equal to negative x cube. I think this is how that will look like. So it will have a concave decreasing slope. So yeah, probably option 4 can be correct. Yeah, so you start from, so if your y-axis is your base quality and your x-axis is your sequence cycle. So at the first few sequence cycles you have very high base quality, and then with the sequence cycle increasing, the base quality kind of goes down, goes down, goes down. So it becomes a curve like a decreasing concave curve, right? So it becomes something like this, something like this. So if you just assume this is the curve that is going to happen. So this curve will have negative first derivative and negative second derivative as well. I think this can be correct. Yeah. Yeah. This question we did not discuss in the assignments discussions. Okay. Okay, then I'll ask this, and I've noted down these questions. So this is one question, and this is the other one. I'll ask and I'll get back to you. So let's see what, which among the following. Which week is this? I think this is also week 1, right? Question number. See, option that option 2 is incorrect. For radioactive DDNDPs, please with. Let me get. Okay. So I can see a separate options here. So just let me show you my screen. these are the options that are shown here. So 4 radioactive DDNTPs replaced with 4 fluorescent DDNTPs. This is my second option. Replaced for carrying forward the reaction. The options are slightly different. I don't have carrying forward the reaction. So the thing is, not sure if the carrying forward thing. Yeah, sure, share. Hmm. Okay, so if you go to Sanger sequencing, yeah, just a second. So the thing is, in terms of Sanger sequencing, we talked about the fluorescent DDNTPs that replaced the radioactive labeled DDNTPs that were there in the Maxim Gilbert method for DNA sequencing. But okay. Yeah, probably. So the thing is, with these questions, the wording. Yeah, I also agree. I think the second option is correct as well. But the thing is, with these questions, the wordings are a little bit have been done in a tricky manner, so that some options become like irrelevant in terms of the questions. So again, I need to. I'm not very sure about this, because I also think the second option is correct, because like, yeah, the Sanger sequencing main part is that you have these fluorescent DDNTPs that actually give out the signals while they attach, and then you can capture that signal to see if what the position, what the read is at that position, right? uh, so I'm also not sure about this. No, acha. I think I got the word. It's not. It's not. It's okay. I got it. It's not fluorescent. The whole idea was to have a DDNTP, not a fluorescent. So instead of radioactive DDNTPs, you had so instead of radioactive DNTPs, there it was not. So in Sanger you could do sequencing by synthesis with a stop, right? So in terms of the first Maxim Gilbert sequencing, you had radioactive DNTPs, not DDNTPs, DNTPs that got attached and you gave you radioactive signals. In the second case, in case of Sanger sequencing, the fluorescence was not the part. Fluorescent came for in terms of Illumina and those kind of things. Fluorescence was not a part of the Sanger sequencing. The DDNTPs were the main part. So you have the DDNTPs, they get attached and they do not uh, allow for further sequencing. So yeah, fluorescent is the issue. Fluorescent is not the thing. So radioactive DNTPs got got replaced by DDNTPs. Yeah, if I just let me check once. I think I'm correct. Yeah. Yeah, DDNTP was the thing, not fluorescent. Sorry, I did not understand. What are you asking about this? I was gotten confused with these. Uh, yeah. Sorry. So I'm not getting it. Like, the issue here is basically the fluorescence, right? Sanger sequencing did not use fluorescence. That is the one that I'm trying to say. The Sanger sequencing used in a host. Let me share my screen. So this is Sanger sequencing, right? So this is the Sanger sequencing protocol, that you have a lot of DNTPs, which are your ATGC normal ATGCs. Then you have a small fraction of DDNTPs, which is like 1 to 99% fraction. Then you add the DNA polymerase enzyme, and wherever the DDNTP gets attached, the sequence gets terminated. After that, replication does not happen. So then you measure the length of each strand in the gel electrophoresis, and then you say ki what. So it will come as bands like these, consecutive bands like this, and then you can get the whole sequence from these consecutive bands, right? So the use of DDNTP was the kind of the main thing in terms of Sanger sequencing, right? The fluorescence came later. Fluorescence came in terms of pyrosequencing. Then I don't think I covered Illumina here, but yeah, Illumina, pyrosequencing, these kind of things. After these, the fluorescence came. Fluorescence was not used for Sanger sequencing. So I think that is the thing that is making it. Yeah. Okay. Anything more? Okay. So I think you spent a lot of time today regarding these. I don't think I'll be able to complete the this is with three, complete all the discussion in term of the assignment questions. I'll quickly breeze through it because I don't think there are a lot many uh, issues with these questions. So just before I move on, so week 4 question number 16, week 2 question number 2, and week 1 question number 15 to 17. These are the problematic ones that I need to talk about, right? If you have anything more, let me know. I'll write in the same mail to sir or to his TM. Okay, I'm assuming that is all. Okay, let's quickly go through these. Yeah, so gene protein relationships that we are talking about there. So the question is, which one is true? So one gene carry one protein, one gene can code for multiple proteins. It can have 1-to-1 or a complex. So this is a much more straightforward one. One gene can code for multiple proteins. that is true. And genotype and phenotype have a very complicated relationship. It's not a one-to-one. The one-to-one relationship exists in monogenic disorders like sickle cell anemia, like cystic fibrosis, and these kind of diseases. But where a single mutation actually caused the particular phenotype. But in most cases, it's not that one-to-one relationship. You have a very complicated relationship. We have polygenic disorders and polygenic traits in most cases. So yeah. So again, monogenic disorders, as I was talking about, a single mutation or single variation causing a particular phenotype. So in this case, if you search these things, these are all monogenic disorders. Cystic fibrosis we discussed, familial Parkinson's. Parkinson's itself it's a polygenic disorder. Do not make that mistake. Parkinson's and Alzheimer's, these are mostly polygenic disorders. But familial Parkinson's disease is caused by a single mutation or a single set of variations that are kind of inherited from the parents. That is there. Huntington is a classic case of a short tandem repeat in chromosome 17, if I'm not wrong. It's a single repeat unit that causes this particular disease, and also the muscle dystrophy disease as well. All of these are monogenic disorders. One variation or one type of variation in a single organism can cause a single human species can cause this particular disease. All are monogenic. If you have any questions, please write in the comments, or have any doubts regarding this, because I'll be quickly breezing through this because we do not have time to fully discuss this. Yeah. Okay, this is a question that I had a particular issue with. Sorry, I think my yeah. This is a question that I had a particular issue with. So 47 chromosomal disorder, it can have. So if you search for 47 chromosomal disorder, it can have both the first options correct. So one is a Klinefelter syndrome, which you have an extra X, and there is also an extra Y, which is the 47 chromosomal disorder. It is termed as 47 chromosomal disorder. So technically both these options are correct, but for the extra Y, so XYY, it is directly termed as 47 chromosomal disorder. So, and for extra X, XXY, for that it is this a known disorder which is known as a Klinefelter syndrome. So technically first option is also correct, but here the second option is given correct. And generally the patients are usually longer and having a bigger size than normal because you have that extra Y chromosome. So that is true for that. So structural chromosomal instability disorders. So basically disorders caused by structural variations in the genome, which are big variations in the genome. And all these deletions, amplification is basically duplication, translocations, inversions, all are structural variations. These all can cause diseases. But yeah, so the answer should be all. But in most cases you'll find deletions, duplications, and are the major causes, but you have examples for translocations and inversions as well. So yeah, can be caused by all. Yeah. Okay, bow tie mapping fails in basically cases where exons are separated by large intronic regions, because again, if you remember the algorithm, that you have one part of the read aligning to one part of the genome, the other part of the read aligning to other part of the genome. So if it is separated by long intronic regions, it becomes difficult for the mapping to take place. So yeah, the answer would be exons are separated by large intronic regions. Okay, before we go into these kind of questions, so there are a lot of these cuff softwares. So there's cufflinks, cuffmerge, cuffdiff, cuffcompare. All of these are legit softwares. So for each particular use case, we use one of these. So when one isoform is present in one tissue but not in other tissue, then you can use cuff compare. So you compare different isoforms and what are their differences, and you say like and then you quantify the the transcript levels of these two particular isoforms. So that is for cuff compare, right? Do you have any questions till now or anything? Yeah. If you have anything, please write in the chat box. If you have any doubts regarding these, yeah. So for comparing different isoforms, you can use cuff compare. For annotating differentially expressed genes, you use cuff merge. So again, so you'll have a GFF or a GTF file where you have genic information and the transcript information, and in your own data set you have the transcript and the quantification information. Kind of merge them using cuff merge. So these are like different softwares that are used for different things. Yeah. So here, capable of RNA splicing is capable of identifying RNA splicing. So different isoforms and alignment in an aware fashion of the presence of introns that have been cut off. So the answer is top hat and star. Again, bow tie has this issue that you have RNA splicing multiple isoforms. Bow tie generally does not work well. So does cufflinks. Cufflinks has other functions as well. But star aligner is the alignment that we talked about, and top hat also I mentioned briefly in the presentation as well. So these two are the ones that are like kind of splicing aware tools that can be used for read alignment. So yeah. Frame shift mutations, again, I don't think I have discussed frame shift very well, but just let me show you what frame shift means. I think the figure should be good here. Some figures here. Come on. Yeah, so you have codons, right? So you have codons. So this is your mRNA, and each of the 3 base pairs will constitute a codon, which will be translated into a protein, into an amino acid, right? But if you delete, say for example, the first base, right? If you delete the first base, the frame shifts. So now the first codon won't be GCU, it will be CUA. The next codon won't be ACG, it will be CGG. So according to the codon, it can if change in codon, it will change the amino acid base that is going to be produced. So in this case, the GCG has been changed with the GAG. So this CGG has been changed to GAG. This particular CG has been deleted, right? If you delete this particular CG, the next codon becomes GAG. Instead of arginine, it codes for glutamine. So these type of mutations are called frame shift mutations, right? So frame shift mutations can cause a lot of things in terms of proteins, and it can cause all these things. It can cause neofunctional, it can cause a loss of function, which is a non-functional protein, subfunction, which is also a variation of loss of function, and basically all of these. So yeah, frame shifts are most deleterious mutations that are possible. So most of the loss of function variants are frame shift or splice variants. So it is quite imperative. Neofunctional frame shifts are rare, but it can still happen, by the way. So elusive genes are basically genes that are difficult to kind of find or characterize, right? So it has very high GC content, it has high gene density, high mutation rate. So basically these are genes that you do not know the functions to, right? So the characteristic of these genes, you do not know what these functions are and hard to characterize. These may be genes that are kind of were lost due to evolution, due to selection, and those kind of things. So these genes, they generally do not have particular functions inside the cell, and they're characterized by uh, high rates of substitution, high rates of mutation, because selection is not occurring against those genes, because that has already occurred, right? A loss of function has already occurred. On top of that, if you do mutations, it's fine. So you have a lot of mutations in these genes. You have high GC content, high density, and mutation rate. So yeah, that is, all of these will be true for these types of genes. So remnant genes are basically a gene that has been part of some mutation, and that has, so for example, we have, humans have been evolved from Neanderthals, and they have evolved from chimpanzees, right? So there are some genes in chimpanzees that are they're present in our genomes as well, but these are kind of remnant genes. These also do not have very high functionality in the genes, because these have, evolution wise, these have lost functions. But you can search it through BLAST, because you can do multiple sequence alignments and genomic alignments in general. So for BLAST, you can use multiple sequences of different species to find which species remnant is this particular gene, and obviously genomic alignment is something that we do for BLAST as well. We do genomic alignment, so that is true as well. Any questions till now? Okay, yeah, if you have any questions, please write in the chat box. But yeah. Okay, we'll not dwell much on this. DEG stands for differentially expressed genes. So the second, fourth option. So DEG is a short form that is used in terms of in RNA sequencing analysis. It stands for differential expressed genes. So yeah, so again, these are all cuff. I don't think the second one exists. This is cuff compare exists. If compare, I don't think exists. But for analyzing differential gene expression, the soft tool to use is cuff diff. Right. Yeah. So again, if you do cuff diff, there's a plugin package for cuff diff to visualize these the differentially expressed genes, which is this particular program, Kamar Band. So it is an R program. Again, this is a plugin to cuff diff. You can use to analyze the DEGs using cuff diff, and then plot the results using this particular plugin, which is Kamar Band. Okay. Yeah, this I'll let you answer. Yeah. No, actually, positive selection will happen if it is happening like a good thing is happening, then the positive selection will happen, right? So if some trait is getting developed that is helping you survive, that then positive selection will happen. Technically, if you are answering this question, the correct answer should be negative selection. So the idea is that the function of the gene is no longer required. So that is why that gene is getting a loss of function mutation, and its function is getting lost in the next generations. But here the negative selection is not given as an answer. The next best answer would be relaxed selection. So the relaxed selection is basically where the selection pressure has been reduced a lot, so that gene is not very important anymore. So then in that scenario, a loss of function can happen in the upcoming generations. Just a second. হ্যাঁ বলো। হ্যাঁ ম্যাম বললেন যে, আমি ম্যামকে জিজ্ঞেস করলাম, ম্যাম বললেন যে, হ্যাঁ শুনতে পাচ্ছ? হ্যালো, হ্যাঁ শুনতে পাচ্ছ? হ্যাঁ ম্যাম আমাকে, আমি ম্যামকে জিজ্ঞেস করলাম, ম্যাম আমাকে বললেন যে মেল করতে টু নারাহারি স্যার ফর দিস, যে আই ওয়ান্ট টু ডু দ্য টেক অন দ্য রেসপন্সিবিলিটি। ও ওকে, আমি দেখিনি, আমি তো পড়াচ্ছি, আমি করে নিয়ে তো আমি দেখে তোমায় কল করছি। ঠিক আছে। ওই তো অ্যানোটেশন, অ্যানোটেশন অ্যান্ড মেডিকেল রিলেভেন্ট, সেকশন S7 S7 S8। হ্যাঁ S7 S8, মানে আমার S7 মোস্টলি অ্যান্ড S8 ইজ প্রিয়াঙ্কা, বাট লাইক কিছুটা ওভারল্যাপ আছে, বাট একটা আমার একটা ওর হলেও হয়ে যাবে, উই ক্যান ম্যানেজ অন আর। ঠিক আছে। হ্যালো, अरे सुनो सुनो, S7 मैं ले लेता हूँ, S8 तुम ले लो, फिर वी कैन लाइक हम लोग आपस में समझ लेंगे, ठीक है? हाँ ठीक है, वो दैट इज़ गोइंग टू बी बेटर। हाँ ठीक है, ओके। Yeah, sorry for that, guys. Yeah, so the idea is that technically here negative selection should happen because the gene does not I- is not that functional anymore, so it is getting negatively selected, so you are having loss of function genes, loss of function variants in those genes. But in this case, the relaxed selection will be the answer because negative selection is not here. And also relaxed selection is basically the selection pressure has been reduced, so it is just a step before negative selection. So in this case the answer is relaxed selection. If you have any doubts regarding this, please write in the chat box or like unmute. Okay. Yeah. The question is which one is true about gene loss, right? So the answer is, first of all, gene loss technically almost always leads to disruption in the genes. ORF is open reading frame, which is the set of codons that is present in the mRNA. So that is the open reading frame of the gene. So it generally disrupts the ORF. Uh, it can occur. Gene loss technically occurs more with the disruption of ORF, but it can occur without the disruption as well. So if there is a substitution in the splice region of the genome of the gene, the splicing will not happen properly, and it can lead to gene loss, right? So not only frame shift variants. So the frame shift variant will technically disrupt the gene's open reading frame. But if you have, say, for example, a splice acceptor or splice donor variant, it is a substitution variant, but it does not allow the splicing of that particular mRNA. So it ultimately leads to loss of function. So that can also happen. So second option is not correct. And locations, remnants of the genes where loss has occurred usually do not show RNA expression. Generally, that RNA, if you have a loss of function variation in the particular gene, generally when that RNA is formed, even if it is formed, in some cases it won't be formed. Even if it is formed, it will be destroyed because it won't be able to produce a functional protein. In some cases, it can produce, but in most cases, it won't. So it won't show any RNA expression. So option 1 and 3 are correct. Yeah, this you can answer, I think. At some level, I think all the options are correct, but yeah. So the answer is, in this case, they have selected the answer that genes under relaxed selection have high prospects to get lost in the future generations. We discussed this in the previous questions, but at a certain level, actually, all of these options are correct, right? So gene loss can occur in those species in which the gene function is not required. This is correct. Pseudogenized gene cannot show RNA expression. A pseudogene by definition does not show RNA expression because it does not code for anything. So that is also correct. Again, the third option is the correct one that we discussed. And the fourth as well, gene loss leads to functional deficiency in the organism. That can be true as well. So at a certain level, all the four options are correct. But in this case, they have went for option 3, which is what we discussed in the um, previous questions. But yeah, at a certain level, this question is a little tricky. At a certain level, all the four options are correct. So yeah, yeah. First option not so much probably, but yeah, the rest of them at some levels are correct. This is straightforward. The answer is BLAST, because you will do multiple sequence alignment with multiple species and you will compare the genic region of this particular insulin gene across different species. Human insulin versus chimp insulin, and you'll try to do it in BLAST. So this is a straightforward answer. This is also straightforward. So all uh, so options 1 and 3. Option 4 is also probably correct, but segmental deletion is not something that we majorly see. Segmental duplication is what we see, but deletions in the gene can lead to gene loss. So a big structural variant in the gene can lead to gene loss, but the other ones are straightforward. Non-synonymous mutation in the exonic region. In the intronic region, it won't like lead to anything unless and until that intronic region is some part of some enhancer or some upstream thing that is helping with the gene expression. If that is the case, then only it is possible, but other than that, in most cases, non-synonymous mutations in the intronic regions does not matter. Regulatory regions, if you have mutations, it will be a problem in terms of expression, and it can lead to gene loss. And yeah, that is there. Okay, final one. For this one, I also am not very sure about the answer, because yeah. I think the options are like all of them. Yeah, all of them are correct. So these are, if you see, there are have been studies done in all of these genes actually to, in terms of loss of function in genes and how it can affect the adaptability. So these are bird specific genes that have been done. Yeah, I think that is all from my side. Yeah, I think we have covered the lot. Uh, if you have any questions, again, go ahead. Uh, yeah. If not, we'll meet tomorrow at the same time, and by then I will try to sort this out, this question assignment issues. I have asked him. I think he will be replying to me. When he does, I will be sending that. Okay, if you have any questions, you can ask. Yeah, I can do that. I can do that in the same. Yeah. So first, to answer the first question, yeah, I think I can do that. I can upload with the correct answers in the same folder where I upload the PPTs. That can be one option. Or if I get it before tomorrow, I'll discuss it in the tomorrow's meeting only, and I'll give you the answers. But I'm not sure. I haven't gotten a reply yet. So let's see. And yeah, tomorrow we'll have a class. To answer your question, tomorrow we'll have a class which will kind of recap the whole 8 weeks. It will be a recap session of all the topics that have been covered before your exam. So yeah, tomorrow same time, 6 to 8. Yeah, I think, yeah. Let's see if I'll try to get the answers by tomorrow's session. If I get that is well and good. If not, then I'll think of. Can you tell me when your exam is? 20, right? So you have a couple of days in hand. Okay, let's see. If I get it tomorrow, that's fine. Nato will think of something. Okay, so week 4 question number 17, week 1 question number 15 to 17, and week 2 question number 2. That's all I'm going to ask. If you have anything more, just send it to me by email, or you can write it here as well. I'll ask them. But if not, then it's okay. I'll close this session. Okay. Thank you for joining. See you tomorrow. You want to say anything?